Hello, everybody. It's now 2 o'clock GMT. Still some participants coming in. Hope everybody can hear me. What I really would love if all the participants who are online could please mute yourselves. If not, and we have some noises in the background, I will try with Henny Groenewald's help to switch their microphone off. So there was already one participant where I switched off the microphone. So again, a very warm welcome to the first webinar of ISBR. We expect more than 100 participants today, and I see on the list we are approaching the number of 100, very close, 97 at this moment. Give maybe 1-2 minutes time that everybody catches up. We will have 1 hour time today. 20 minutes for first speaker, 20 minutes for the second speaker, and then 20 minutes for discussion. Of course, not everybody will have the time to ask a question. Maybe you can also use the chat function. Okay, so we are now with the number of 109, so I will start. So again, warm welcome. My name is Detlef Bartsch. I'm President-elect of the International Society for Biosafety Research. And I will have 4 slides in the beginning to introduce you to our webinar. It works. Okay, so as I said, we are topic-based in this webinar series with 2 times 20 minutes presentations, 20 minutes discussion. We try to focus on current topics relevant to ISBR members, and we invite in our webinar series 2 speakers that can give different perspective and complement or contrast each other's. And very important for you, the meeting will be recorded and published on YouTube afterwards. The background of our first webinar is the brand new, more or less, EU-Mercosur partnership agreement. It's a trade agreement, but has also some impact on biotechnology. You see here on the map, in orange, the Mercosur member states, and 2 of them, Bolivia and Venezuela, are not parties of this trade agreement. And here in green, the EU member states. Just recently, 26th of February 2026, Uruguay and Argentina ratified the agreement, and 1 day later, the Euro Commission announced that it will provisionally apply the agreement. And if you would like to know more about this trade agreement, the Wikipedia is not bad, I must say, and with a lot of links and close to the actual state of the art. Article 7 of the Mercosur Agreement establishes a framework for cooperation on agricultural biotechnology, focusing on managing asynchronous GMO authorization, the low-level presence of GMOs, and information exchange. And that was the reason for why we choose this for today's webinar on data requirements. But take into account, it's the ISBR focuses on biosafety data. I will repeat this at the end of today's webinar, but just to let you know that this will only be the start of a series of webinars. The next is planned in June 26 and followed by some others. We would be very happy if you could send us suggestions for more webinar topics to this email address. I will repeat this again at the end of today's webinar. Okay, having said this, I now show my full face and would like to hand over to our first speaker, Katharina Unkel, who formerly worked at the German Federal Office of Consumer Protection and Food Safety, now joined the Robert Koch Institute, but she will give us a more European EU project perspective on data requirements. So Katharina, you have the word, please.
Yes, thank you. Welcome from my side as well. I'm gonna share my screen. Yeah. So thank you, Detlef. In this talk, I want to give a short overview over the, I think I have to change something. Proposal for a new regulation for plants bred with certain new genomic techniques. And I will also show some of the work we did within the GeneBEcon project. But first, a little disclaimer though. My contribution does not represent an official opinion of the German government, and I have no conflict of interest. I'd like to start with some points about the European Union, where we are 27 countries that share one single market with free movement of goods, services, capital, and people. And with one single market for 27 countries, there also comes shared legislation. The ordinary legislative procedure follows the trilogue between the Council, which represents the 27 governments of the EU countries, the Parliament, that represents the citizens, and the Commission, that represents the overall interest of the European Union. The Commission provides a proposal for new legislation, which is then discussed within the trilogue. And if they come to an agreement, the Council and the Parliament then officially adopt the new legislation. One of those European legislations is the genetic engineering law. Here we share one framework for GMOs, and in this talk I will talk about plants. So for plants, this shared framework covers the field trials, cultivation, and import of GMO plants. It also defines what a GMO is, and here it says that it is an organism, with the exception of human beings, in which the genetic material has been altered in a way that does not occur naturally. However, since this definition was formed, new techniques have been developed. Those techniques, the new genomic techniques, or NGTs, are capable to change the genetic material of an organism and have emerged or have been developed since 2001, so after this law has been established. For targeted mutagenesis can be achieved by a variety of different techniques. Here they are zinc fingers, and I have to get it right, transcription activator like effector nucleases or talons, and mostly known probably the CRISPR-Cas technology here on this picture. It is together with the Cas9 endonucleases. So all those methods, though, have in common that they lead DNA cutting enzymes to a targeted site on the DNA where they induce breaks, and in those breaks, the genetic modifications take place. Another NGT method is cisgenesis, where genes from conventional plants can be inserted, or they can substitute endogenous genes, but they can also be inverted and translocated within the NGT plant. Okay. And in 2018, a decision was made that those techniques in plant breeding would always lead to a GMO, and that those GMOs would fall under the restrictions of the European genetic engineering law. This gave reason for the Commission to do a study on NGTs under the Union law, where they came to the result that the current engineering law, genetic engineering law, is not fit for purpose for plants resulting from certain NGTs. And this result laid the base for the proposal of a new regulation. And as of now, there has been a provisional agreement within the trilogue between the Commission, Parliament, and Council. And now it must be formally adopted by the Council and the European Parliament, which is expected to happen in the spring of 2026, so this year. It will then be published during this year and start to apply 2 years later. There are, in the proposal, NGT plants are categorized into 2 categories. In category 1, NGT plants are equivalent to conventional plants. There is a verification procedure based on objective criteria. They are subjected to the rules applied to conventional bred plants. Their seeds are labeled as NGTs, and information must be available in public databases and variety registers. There will be no risk assessment for those plants and also no risk management. On the other side, we have category 2 plants, which are not equivalent to conventional plants. They follow an authorization procedure with adapted risk assessment, and detection methods are required. They are traced and labeled as GMO with a voluntary statement on the purpose of the allow for certain incentives for NGT plants if their traits are desirable. They are following coexistent measures, and there is no opt-out. There will be case-specific risk assessment and also case-specific risk management. The whole plan will be monitored and reported on and will then be revised by the European Commission. For now, NGT plants are prohibited in organic production. Coming to the criteria of the equivalence. So plants obtained by targeted mutagenesis can't have more than 3 genetic modifications per protein coding sequences. The substitutions and insertions that took place are not allowed to be more than 20 nucleotides, but deletion of any number of nucleotides are possible. For cisgenesis plants, the combination of DNA sequences must come from the gene pool for conventional breeding purposes. There's no interruptions of endogenous genes are allowed, and it is not allowed to lead to chimeric proteins. For all NGT plants, the genetic modifications in any possible combinations can't be more than 20 per monoploid genome. Excluded from category 1 are NGT plants with new traits for tolerance to herbicides or which produce a known insecticidal substance. Coming to the actual data requirements, so the biosafety data, it starts with name and address of the requester, designation and specification of the NGT plant, a description of the trait and characteristics that have been introduced, and also technical information, which means studies relevant to the DNA sequence information, information on the use technique, and information that the plant does not contain foreign DNA when such DNA have been inserted during the development. There are also the requirement to declare that the NGT plant meets the criteria of the equivalence, a declaration that the intended traits are not a tolerance to herbicides or the production of a known insecticide substance, And also the requester should say what information should be treated as confidential. Scientific evidence is asked for to show the relation between the introduced genetic modifications and the intrinsic traits. All these data requirements can be changed by the Commission if there are new information or during the reporting process, or the reporting process gave reason to change those requirements. For the category 2 NGT plants do follow the comparable approach for classical GMOs in their risk assessment. This covers molecular characterization of the plant. Also, there's a comparative analysis of compositional, phenotype, and economic properties, so field trials. There is safety assessment for humans and animals about allergenicity, the toxicology, and the nutritional value of the plants. And there's also safety assessment for the environment. However, for classical GMO, there is not really a case-by-case approach, but there's mainly full data sets required. But for category 2 plants, the type and amount of information will be adapted case by case. And these decisions are influenced by the traits that have been introduced, by the function of the inserted genes, and in case there have been disruption of genes, their function is of importance as well. Prior experience with the cultivation of similar plants and prior experience with the consumption of similar plants or products, the scale and condition of the release, as well as the intended conditions of use, and also the potential receiving environment. This brings us to the GeneBEcon project, an Horizon project that ran from September 2022 to September last year with 18 partners from 11 countries. And it aimed to examine the innovation potential of gene editing in enabling sustainable bioeconomy in Europe. Part of that was the development of 5 possible regulatory options for NGT plants and their biosafety data requirements. Here we have the status quo, so the current legislation labeled as option 1. We have option 2 with case-specific adaptations to the status quo. In option 3, it was important if the character of the new plant would be conventional-like or not conventional-like. In option 4, only the trait itself was of importance and its novelty, and not the genotype of the plant. In option 3, it was only of importance of the plant was free of foreign DNA. And in option 6, it was REACH-like, which is the regulation on the registration, evaluation, authorization, and restriction of chemicals. So going to another field. More information on these options are available in a published paper that you can look up if you like. And comparing it to the new regulation of NGTs as proposed by the Commission, option 2 would be close to category 2 plants with an adapted risk assessment, otherwise following authorization of classical GMOs. And category 1 plants would be treated similar to plants of option 3, which are found to be conventional-like. Since the project had the bioeconomy in mind, we did look at the possible costs for those data requirements. The cost for option 1, or the status quo, was mainly taken from a study of the European Commission on the impact of the legislation of NGT plants, which has been published in 2023. We set those costs at 100%, which was at a maximum around 60 million euro, concluding costs for field trials, rodent feeding trials, biodiversity assessment, and so on, and compared our options to those 100%. And so for option 2, in green, you can see the amount or the cost of the verification data, which was collected in a first step to confirm the status of the NGT plants and to define the biosafety data needed. So in option 2, according to this verification data, field trials or feeding trials, which are a huge cost factor, are no longer mandatory, which is why you can see this gray cascade. For option 3, the verification data is thought to be data that has already been established during the research and development phase of the plants and is mostly molecular data as well as some literature search. And for some plants, the cost to collect those data was as low as 3,000 euro. And here as well, for more details on those results, you can check out the deliverable, which is a report on biosafety data requirements. You can visit the GeneBEcon website by checking the QR code. And soon, we hope this year, there's another paper coming out where which is about comparing the effects of regulating new genomic techniques on the investment decisions in the European Union, where one of the big findings was that investment decisions are highly influenced by regulatory uncertainty as well as the authorization procedure itself. So with that, I would like to thank you all for listening and looking forward to all the questions that will come later.
Thank you very much, Katharina, also for taking exactly the time of 20 minutes. So fantastic. And so be patient for taking questions after the next talk. So with having said this, I welcome Marcos giving the second speaker, giving the perspective of Brazil in this respect. Marcos, are you ready?
Yes, and thank you. Good morning, good afternoon, and good evening for everybody. Just going to start to share my screen here. There we go. My name is Marcos, and I'm Director of Regulatory and Scientific Affairs of the Brazilian Association of Innovation. As we have seen by Katharina, gene editing is rapidly transforming biotechnology, agriculture, and food innovation. Brazil and other Latin American countries have implemented clear science-based regulatory frameworks that balance innovation with robust safety oversight. Brazil, together with other Latin American countries, stands out for its case-by-case approach, providing regulatory clarity for gene-edited products that do not contain recombinant DNA. So across Latin America, authorities are increasingly aligned with international best practices, creating a predictable and innovation-friendly environment. This regulatory maturity positions the region as a relevant and forward-looking hub for the responsible development and commercialization of gene-edited technologies. In Brazil, the biosafety law was published in 2005. That was the second one. The first one was published in 2000. So in Brazil, we do have more than 25 years of biosafety regulation. And even published at that time, the regulation was guided by the promotion of scientific advancement in the field of biosafety and biotechnology, and of course, the protection of human, animal, and plant life, and also for the protection of the environment. And also in Article 10, it says that CTN Bio, that is the entity, legal entity in Brazil, that is responsible for all GMOs, including gene-edited evaluations, shall monitor developments and technical and scientific progress in the fields of biosafety, biotechnology, bioethics, and related areas. Okay. Having said that, in 2008, Brazil has had the first regulatory norm of regulation for commercial activities for releasing GMOs in Brazil, and then was updated for a new regulation in number 32. In 2012, with the emergence of CRISPR-Cas9 technologies, enabled genome modifications to be performed faster at lower cost and with greater safety. A regular GMO usually is done for just a few multinational companies. It's very costly, very time-consuming. So with this and other techniques, with this new one, so enable the possibility for even national or smaller countries, companies, or even public companies to do such modifications. So in light of this new emergent technology, CTN Bio established a working group to technically evaluate products developed using new breeding techniques. So, and of course, and how to determine how they should be classified under the biosafety law, and it was required to have a new regulation. So after 3 years of discussions, CTN Bio is composed by 54 members, so 27 are full members and 27 there are alternate members. So they had 3 years for a let's say a long discussion regarding NGTs, which include gene editing technology. So by 2018, a specific regulation was published regarding NGTs, gene editing, which implements a case-by-case. So it's very important to highlight it. Case-by-case consultation system to determine whether a product developed through an NGT should be classified as a GMO under the Brazilian biosafety regulation. Some information that should be provided by the developer, such as organism of origin and the developed product, method used for product development, molecular characterization of the product, and the criteria for classification as a non-GMO conventional one. So a product may be considered non-GMO if it meets one of the more of the following criteria. The first, probably the most important, is absence of recombinant DNA, RNA. Presence, for example, of genetic elements that could be obtained through conventional breeding. Presence of induced mutations, such as chemical mutagenesis or even radiation. Presence of mutations that may occur naturally. Here it's an example of required study packages. For example, for a commercial approval of GM microorganism in Brazil that is regulated by RN21, compared to a consultation letter of RN16 gene editing. So for molecular characterization, should provide lots of information. For example, for a consultation letter, description of the technique used, for example, CRISPR, and of course, proof of the absence of foreign DNA, and the conventional one, GMO, protein analysis, food safety, food feed safety, composition equivalence, environment assessment. So for a dossier, just for you to have an idea, at least here in Brazil, the size of the dossier for conventional GMO for a microorganism could be 300, 500 pages. For a plant, it could reach up to 1,000 pages, while a consultation letter may reach only 50 pages. So it's much simpler situation. Here is the workflow. So the interested party should make a submission to CTN-Bio. So this is publicized in the official gazette, so everyone can have access to this information. It goes to CTN-Bio members, that 54 members that I mentioned previously to you. So they had 90 days, and of course, if it's necessary, it could be extendable. And then the two subcommittees, human, animal, and plant and environment, they provide an opinion that goes to the plenary decision when the two subcommittees, they get together and they take a decision if the product is considered as a conventional one. So it goes to the Ministry of Agriculture, for example, for registration, if it's a plant, for example. If the decision take into account the information provided in the letter of consultation, if it's considered a GMO, and then the company has to submit the full package of the information. Here, just a quote for an article that I've seen. I think it's worthwhile to share with you. So to avoid creating obstacle to technological advancement and at the same time to ensure biosafety parameters, updates and adaptations to legislations need to be made to accommodate the effects caused by emerging technologies and to make safety requirements proportional to risks. Here is as an overview of the products approved since the regulation was published in 2018. As you can see, microorganisms in plants are the majority. There are some animals and other products. Here you can see the products where they are used in fact. So, for example, microorganisms, they're being used for editing yeasts for ethanol production. So this is a very important product in Brazil. They're using cars and other things, biofuel, and also for example, for nitrogen fixation of soybean. 85% of soybean in Brazil, they use inoculants. So it's something very, very important for agro-bio products. Also, there are other ag bioproducts that also use microorganisms that were gene edited. And also there are some drought soybeans, drought resistant soybean, herbicide tolerance, high content of sucrose in sugarcane, and even increasing of protein in fish. So there is a very wide range of products that we already use in Brazil. Also, I would like to share with you an overview of gene editing in some Latin American countries. So evaluation case by case is done by Argentina, Brazil, Colombia, Ecuador, Honduras, Paraguay, and Uruguay. Just highlighted Argentina, Brazil, Paraguay, and Uruguay. They're part of Mercosur trade agreement with Europe. And also there is no restriction for example, for Costa Rica, Chile. There is ongoing debate in Cuba, Guatemala, El Salvador. There are some prohibitions, for example, in Bolivia, Mexico, Peru, and Venezuela. Here, I would like to just to provide you some, in case you are interested to look for the regulations where they have gene edit regulations. Argentina was pure pioneer, not in Latin America, but I think all over the world, where they published the first regulation for plants. Later on in 2021, they amended for microorganisms and animals, followed by Chile in 2017, Brazil and Colombia in 18, other countries in 19, and finally Uruguay in 2024. Here is also an overview of gene editing plants in the world. So there is a mix of allowed and not allowed, or ongoing discussion or prohibitions. So it's a technique regulation that is ongoing. There are ongoing situations. So just to close up my presentation. So Argentina, Brazil, Chile, Colombia, Ecuador, Guatemala, Honduras, Paraguay, and Uruguay adopt very similar regulatory criteria for gene editing. In general, in these countries, the institutions should present their products derived from new technologies of gene editing to an advisory body, which will attest that there is no foreign genetic material in the new trade. So with that, I close my presentation, and I'm open for any question that you may have. Thank you.
Thanks a lot, Marcos, for sharing your slides. I think it worked well. We have now 20 minutes for discussion, and also thank you, Marcus, for taking exactly a little bit less of the time. If you have questions, please raise your hand electronically. I can't see all participants, I must say. I've seen that we have now, we had in between 187 participants, a little bit down by 181 now. And there is already one hand I see from Ben Durham. Please, Ben, switch on your microphone and your camera.
Right. From South Africa, good afternoon. Thank you very much to ISBR. Fantastic webinar. Please keep it up. I will make some suggestions for further webinars, but this is a very relevant one from a government policy perspective. Thank you very much. Three questions, please. To the European system, I note that there was a comment about excluding pesticides and herbicides from category 1. Can that be explained a little bit more? What does it mean by excluding them? Does it mean they go into category 2 kind of considerations? But if you can do that. The second one is I think in category 1, they also have to demonstrate similar nutritional value, but there is the possibility of a starch-enriched sorghum, for example, which will have deliberately which will deliberately have a different nutritional characteristic. Would that require a movement to category 2? And then the last question for both Brazil and the EU, and perhaps I missed it, but is there a requirement to check for non-target modifications? In other words, in CRISPR-Cas, there may be similar sequences elsewhere in the genome, which may then have a similar kind of modification. Is there a need to check for those under the category 1 thing? Thanks very much. I'll listen into your responses. Thank you.
Katharina, I guess the first two questions goes to you, and then Marcus. Please, there are two others in a row, so please not.
I try.
Okay.
I try to keep it short. So the first question was why traits for herbicides and the insecticidal proteins are taken out of NGT 1 category. And the reasoning behind this is that Europe follows big plans on being as green as possible and as green as possible. So the idea behind this is that if herbicides resistant is introduced into the plants, but the regimen of treating plants with herbicides isn't done according to those plants, it might lead to resistances in the wild plants. So this is why it was taken out of the NGT1 category, just to be sure that it wouldn't lead to an increased use of herbicides, because Europe is following the road of trying to keep the usage of those as low as possible. And similar idea is behind the insecticidal substances produced in the plant, just to make sure that it might not affect non-target organisms. Since for the category 1 plant, there is no risk assessment or no risk management plan. So any traits that might go into this direction are included from category 1. So that's the first question. I hope that was short but sufficient. And for the composition questions for NGT1, they are not, it's not about composition information. It's only the criteria of equivalence. So they wouldn't even ask for that. As long as it's not, if it's within the limits of those criteria, it wouldn't matter. So if it would be NGT2 plants with a major change in composition, it would always be just looked at and assessed accordingly. But yeah, there's no need on those data for NGT1.
Before I hand over to the Marcus part, I must say that it's a little bit inconsistent, and when it comes to equivalence, that herbicide tolerance is excluded from tolerance from category 1 because with mutation breeding you can also generate herbicide tolerant plants. So it's a political thing in Europe to get say the support of also parliamentarians.
Yeah.
Okay,
it's good that you say that and not me.
Okay, no, it's a compromise.
You can say
so that's purely political reasons, to my opinion. So, Marcus, please.
Yeah, thank you. Thank you for the question. And for what the regulation says, it should be absence of target. So it should be analyzed to be sure that there is this absence.
Would maybe add that during the implementation phase of the EU regulation, we have to figure out what extent of data we need to have to show that there are no target alterations or little target alterations. And I guess that informatic information will have a certain role as well. So let's see how it develops. So I see two other questions. First one is German Vigi. I hope I have it pronounced. I remember you. German, please.
Thank you, Detlef, and thank you very much for the nice webinar. I have one question for Marcos. I was looking at this very interesting paper with all the technologies obtained with NBTs. Do you know how many of them already reached the market? I mean, and when authored the authorization request process?
Yeah, there are. I don't have this number here with me. I can check it later on. But the majority of the products they are already on the market, including, for example, for yeasts, for sure. Lots of them are on the market, but I can try to see which one are already being used and share with you later on. But it's a technique that has been widely used here in Brazil.
Thank you very much, Marcos. Thank you.
Okay, I saw that there's also a question in the chat, but before I will address the question by John McMurdy, I will hand over to my dear friend Salvatore Apaya.
Thank you very much, and thanks for the webinar. This was quite a nice update. I have a question for Marcos. I noticed that among the criteria that you explained that can be used to exclude any NBT event from being considered GMO, there is one that says mutation that can occur naturally. What exactly that means? Because any mutation could occur naturally. Can you explain a little bit that? Thank you.
Yeah, I will try. I'm not expert in genetics, so I do more with regulation. But so far, what we have seen, it's natural mutations. For example, a cattle that has long hair. For example, you have a natural mutation for having a short hair. So, for example, this cattle can be used in Brazil. It's a tropical country, so it's the animal will not suffer as having a long hair. So this is, for example, an example of natural mutation that has been used. There is another example, but it's drawn later on, that was the size of the horn for a cow in order to facilitate the manipulation of the animal. So naturally, there are some mutations that some animals could have a short horn, and this kind of mutation could be considered natural mutations. There are some examples that I have to share with you. I don't know if it was enough clear.
Hope so at this moment. So, Marcus, can you have a look on the question by Ray Dobbert or the disagreement in the chat? You have a few minutes to think about it if you see it. Would come to John McMurdy, who wants a specification on whether this category 1, category 2 legislation would apply only to cultivation or also to the imports. And maybe Katharina, you can help me. I think it's we do not distinguish between import and cultivation. It's the NGT regulation in the EU is planned for everything. Immediately leaving us into trouble if other countries, and we have this seen in this Article 7 of the Mercosur EU trade agreement, that there needs to be an exchange because we know that there are not only asynchronous authorization, but different legislations and different handlings of NGT organisms. Yeah, that's one part or one big problem, specifically if you don't have sufficient detection and identification methods, which I think will be the case for many products. Right. Okay, but Marcus, have you seen the second question?
Yes, I can check that. Probably my statement is not fully correct. So, but I'll try to find out how many products are on the market using a gene-edit products reviewed by CT. And thank you for Dobert for raising this one.
So again, I'm really happy that you are so disciplined, raising your hand and everything. So wonderful. So the next in row is Louis Luc. Pronounce it right, Louis. Switch on your microphone and your camera, if you like.
Yeah, you did. Thank you very much, yeah, so on that point on the herbicide tolerance and disease and insect resistance, so if it's included for importation, so what is the process between now and the implementing regulation to try to get those out so that the EU can be in alignment with other jurisdictions? So from your perspective, and I don't know if this is a question for you or like for the first speaker, or like if both can tag team to answer this, but what would be a way for this to move forward, or just to actually limit it to cultivation within the EU?
Katharina, would you like to answer first?
Sorry, I was answering a question in the chat. Could you please repeat your question?
Sure, Katharina. Yeah, absolutely. So if it's as it was previously said, it appears that it's actually including imports as well as cultivation for herbicide tolerance and disease resistance being categorized or being put in the category 2 thing. So if that's the case, and if it's political rather than actually or what, then how can we or what's the process from your perspective to get both disease resistance and herbicide tolerance out of that category 2, so the EU can actually be in alignment with other jurisdictions that have issued guidance on these traits?
Yeah, so I can only really give my my opinion on it, because I'm not an expert in the import and I'm not quite sure how it will be done, because comparing it at the regulation now on GMOs, it's quite excessive, the data that is required for GMO imports for food and feed. So yeah, this is a general question. I think in my opinion, the EU hasn't answered quite clearly yet how to deal with the import of NGTs with all those different regulations. I think the idea is to have kind of a fact sheet coming with the plants, but if they stick to their own rules, imported NGT plants with those traits would have to undergo those assessments. I don't know if then the EU will say that they will do it, so they have the cost, or if the importer will have the cost. So to my knowledge, it's not yet really clear how that will be done. Might be waiting for the implementing regulation that might come soon. I know. So you're right.
Yeah,
Basically.
and I feel it's somehow unsatisfying, but it's we are living now with 40, 50 years of GMO risk assessment and biosafety data interpretation and use, and it's not always easy to see the transition of good data we have, and the practical implementation of and use of innovation technologies. I see that Henny Runewald, you are looking at the time, maybe the last one contributing with a question or statement.
Quickly, 3 things. The first one is that there are some questions in question and answers as well, which we can maybe quickly look at. And then I see already there Simon just put in a point there as well about the off targets. I think this is one of the big issues. My question with that is, how much is enough? And the point that Simon made here is actually one that I also like, genomes are not stable. So if we do conventional breeding, phenotypic screening is actually an indirect way of checking that. So I would like to hear other people's comments on that. And then the last bottom line question is, after this discussion in the agreement between the EU and South America, do you think there's enough alignment in the data requirements that you would be able to trade without issue? Thanks.
There are a number of questions, Henny. So who would like to answer? Marcos, Katharina? I could start maybe, because just to mention that before my retirement was also a regulator for the German government, so also involved in these negotiations and interpretation. That it's not fixed yet, and the next 2 years will be very important to see how we address these questions. And as we have seen from the GeneBEcon project, you can clearly restrict the use of innovation by putting the threshold or the level of biosafety data requirements so high that it can't be used or hardly be used, and only by the big companies. And what I learned from Marcos is that small and medium enterprises, even universities, if released from the big requirements, they easily have access to applications on the market. Right, Marcos?
Yes, and also regarding the question, Dr. Nepomuceno just also clarified regarding commercial products being on the market, informed that microorganisms, many of them are already on the market, plants not yet. So, but regarding the that data package of information or regulation, it should be discussed among governments. So it's something that we apply here. It seems to be different, and it should be evaluated for in a case by case situation. So, but I think it's something that Mercosur and EU should start a discussion in order to reach a common ground for both.
Arina, anything to add from your side?
Oh.
Look like you are answering the chat again.
Sorry, yeah, sorry. No.
Okay. Then looking at the time, so we have now 4 o'clock. I will use the opportunity again to present my last slide. We would welcome if you have suggestions for the next webinars. And my feeling is that the current webinar was quite a successful start with only little technical issues, and really impressed how many people were in the webinar. So please send ideas to this contact.isbr@gmail.com address. And yeah, I hope with that, see you all, or even more than for the next webinar. And also very important on the preparation for the next big conference in 2027, October, November. More likely what I heard October than November in Indonesia, on Bali, the next big ISBR meeting where this is extraordinary exchange of ideas and networking possible. So with there, we are also collecting ideas for sessions and everything. So please have a look at the ISBR website for announcements and anything else. So yes, what else can I do? I think I can now close the meeting. Thank you very much to Katharina, to Marcos for the presentations. They will be certainly also separately be available, I guess, somewhere on the ISBR website. We are experimenting for this also. And also saw in the chat that maybe the recording was only possible the first 30 minutes. I hope this was not true, but maybe next time everything is fixed. Okay. Then the link will be sent. I see is a record one have a link. Yes, somewhere we will put it on YouTube. What I remember from our pre-discussion, and we will let you know. Okay, then have a good morning, good afternoon, good evening, wherever you are in the world. Thanks and take care.
Yeah. Thank you as well. Bye.
Thank you. Bye-bye.